Here we've got a culture of yeast, and what we want to do is to take some of that culture and to put it onto an agar plate, and we're going to use our inoculating loop. Now, you've got a loop there. Check that the top of the bottle is not too tight before we start.
Yeah.
And then what we're going to do is we're going to take our loop and you're going to hold it at about a 45-degree angle in the top of that Bunsen. That's lovely. Work it gently out so that it gets nice and hot. Now hold it to the side of the Bunsen for a moment because it's too hot to use immediately. Take the top of the bottle, hold it in the little finger and turn the bottle. Now remove the top, flame the neck of the bottle and then run the wire along the top of the glass just to make sure it really is cool.
Yeah.
Gently stroke the surface so you've got a little bit of the culture on the top. Flame the neck of the bottle, return the top. Now what we need to do is to place that culture onto the Petri dish. Lift up the base and gently streak the loop across the surface. Because that's still contaminated, good, we need to flame it. So again, gently draw it across. And any liquid or any culture left in that loop would vaporize before you actually get it into the hot flame. If you were just to plunge it in, it gets so hot, we get a lovely hiss. Kids often like the hiss. But again, we might atomize into the air. Now, that's a way of doing it. But there is a problem. If you're teaching this to pupils in school, quite young pupils, and they've been listening to you and they've watched what you've been doing, they say, well, you opened the plate, you exposed it to the air. And what we've said to them is, of course, there are microbes in the air, there are spores in the air, and they're going to say they might drop in. So I've got another way of doing it. Again, I check that the top is not too tight. I hold the top in my little finger and I turn the bottle. I flame the neck of the bottle. I sterilize the loop so it gets nice and hot. I wait for it to cool for a second. I run it along the top of the glass just to make sure it isn't too hot. And just like you, I'm just going to gently stroke the surface. Not digging, not mining, just stroking the surface. Flame the neck of the bottle, return the top. And this time, I'm just going to open the lid a little so that I can now just streak across the surface like that. I'm keeping my arm fairly low, my hand fairly low, and just gently going across. When I've finished, I just gently pass the loop through the Bunsen until it gets red hot. And if it's hot and I'm worried about the surface, I just leave it like that.
Okay.
This way, I was protecting so the kids get the idea that things won't fall down into the agar plate. It doesn't matter which method you use, provided you don't get contamination.
Yeah.